Okay, I think we should probably get going. Welcome, everyone. I'm Steve Monk. I'm the Deputy Director of the Biodesign Institute here at ASU and the host for tonight. Thank you very much for joining us this evening. We have a couple of housekeeping items to mention here. There are virtual menu features. You can rename yourself on the Zoom screen if you hover over your name and make a change. Please use the chat box for questions. During the presentation, please mute yourselves for the presentation, but we encourage you to leave your cameras on. So if you have questions or comments, please send those in the Zoom chat and we will address those in the Q&A session at the end. The Biodesign Institute is an institute where science is inspired by nature. So nature has really solved many of the world's great challenges. For example, nature has given us an immune system. And Stephanie Forrest, a computer scientist here at Biodesign, inspired by nature, is developing software that behaves as if it has an immune system. It resists viruses and other bugs and can heal errors in the code. So not biology, but inspired by nature. Our science is published in the best journals, but also has a goal to translate that great science into the public good. An example of that, which I'm sure you're all aware of, is our ASU Biodesign COVID Testing Lab, ABCTL. That is based on research and technology developed by our executive director, Josh Lebert. Prior to COVID, Josh and his team were developing high throughput tests for a variety of conditions, including breast cancer and radiation poisoning. But within weeks of the pandemic being declared, the team pivoted and set up a saliva-based testing in a now CAP, College of American Pathologists certified laboratory. So that's the highest level of certification one can have in a clinical setting. And at this point, we've run well over 1 million tests. I think the last number I saw was about 1.4 million tests in the 2 years of the pandemic. Next slide. Sip of Science is a very pleasant way to learn about our great science in a social setting. This is the 4th year of this award-winning program. It was named the best way to drink and learn in the Best of Phoenix in 2020 by Phoenix's New Times. Sadly, for now, we are obviously continuing with virtual format. All right, so it is supposed to be a sip, and I again have the pleasure of being your sommelier tonight. I'm not a mixologist, so even though it is Mardi Gras, I leave the hurricanes and other New Orleans classics to you, and I will stick with wine. As you might remember, I have a long-standing love of wine and mostly consume the French stuff. While I love Burgundy, for tonight's white, I've selected a Sancerre. Although Burgundy is fantastic, some food works better with wine from other regions. I'm a big fan of shellfish, oysters, and steamed mussels, for example, and I find the wine of the Sancerre region to be the perfect pairing for shellfish. It's a Sauvignon Blanc, but a very unique one in my view. You will not see the word Sauvignon Blanc anywhere on the label. European labeling regulations are much more concerned with the region, the terroir, than the grape. So all you see is a Sancerre. Sancerre wines tend to be citrusy, mineral driven, and have a flintiness. This is in sharp contrast to New World Sauvignon Blancs, say Napa or New Zealand, which tend to be much more grassy and tropical fruit driven. For the red, let's look at the Cotes de Rhone. So the Guigal family has been producing wine in the Rhone region for 3 generations and owns some of the finest vineyards in the region, many costing in excess of $500 per bottle. The Rhone is just south of Lyon. You see the city of Vienne at the top of that map. That's a few kilometers south of Lyon, and it stretches all the way down through France. The Cotes de Rhone is an excellent example of the region's product and typically retails for under $20 a bottle. It is a wine that I've consumed over decades and has been very consistent over the years. It's a blend, mostly Syrah, which the New World refers to as Shiraz, and generally blends with Grenache and Mourvedre grapes. It has flavors of black fruit like plums, cherries, hints of vanilla, some nice spice, and soft tannins. The wine pairs very nicely with lamb or other more gamey, higher flavored meats like duck. So I encourage you to try these and enjoy them paired with food. Now for the main event, the science. It is my pleasure to introduce Efrem Lim, a virologist here in Biodesign, an assistant professor at ASU in the Center for Fundamental and Applied Microbiomics. His lab studies the interaction and evolution of the human virome in the development and immunity of systems. Lim began studying the human virome long before the arrival of SARS-CoV-2 and the global pandemic. Lim and his team have led research efforts to understand and prevent SARS-CoV-2 transmission, along with tracking the variants of concern in real time using next-generation sequencing facilities found at Biodesign. This information is used to inform public health responses and policies. So the title of Efrem's talk, Watching for What's Next, Sequencing SARS-CoV-2 Variants. Dr. Lim.
Thanks so much, Steve. And hi, everyone. Thanks for joining us tonight. What I'd like to do tonight is to share with you how we track the evolution of SARS-CoV-2 variants through the COVID-19 pandemic. And I thought I would start first with sharing you a little bit of my background and kind of the motivation behind our research. Next slide, please. So I grew up in Singapore. It's a small country in Southeast Asia. In 2003, we faced a deadly respiratory virus called SARS as the first SARS coronavirus. So I experienced firsthand the panic created by a new deadly virus. We did not know a lot about this new virus when it first emerged. The picture on the left shows you these tents that we set up outside a major hospital in Singapore. And that was how each patient was triaged. When it came in here, they assessed, did they have SARS-like symptoms or not? There was not much knowledge on how to handle these patients. We did not know how deadly it was going to be and how transmissible the virus was. We never got a diagnostic test like we have right now. If you were at ASU, you can just drop off a saliva test and got tested. That never existed. And to the end of that outbreak, we never got that. We didn't get a vaccine developed for SARS either. And so, and in fact, we took months before we actually sequenced the first genome of this SARS virus. On the other hand, I also experienced firsthand how we can live through a deadly outbreak like that and pull through from that. And so that motivated a lot of my passion in science moving forward. That was one of the reasons why I spent the rest of my career working on viruses, trying to understand new viruses that may be causing human health problems. In my postdoctoral training at Washington University, I in fact joined a lab that was responsible for the sequencing of the first SARS virus. From there, I developed a technology called next generation sequencing in order to study all viruses in the human body. Next slide, please. And so in 2017, I joined ASU, and that's what my lab studies. We study all the viruses in the human body, we call that the virome community. And we're really interested in using this knowledge to improve the lives of mother-infant health, because infants are the future of our generation, of our next generation. That was the main thrust of our research until COVID-19 hit us in 2020. Next slide, please. So in early January of 2020, the first reports of this new coronavirus emerging from China and actually showing up in Arizona state hit us close to home. And this wasn't just some foreign virus, this is right on home ground. And you can see, based on my history, why I thought this was really important to use our knowledge and our expertise to tackle this important problem. And so we were uniquely poised to use sequencing technology to understand how this virus was evolving and to track this virus, because this is a totally new virus out there. Next slide, please. So a little background on genomic sequencing. What we do is we first, for example, if someone is sick, and if you don't know what that is causing you to be sick, what we do is we typically collect a sample, and then we would do a testing on that to figure out what you're sick from. In this case, we can actually take the virus genome from the sample and sequence it using this technology. And on the right, you can see it just reads it out. Each alphabet on the genomic blueprint of the virus is going to be sequenced out. This tells us what virus it is, what variant it is, what mutations it has acquired that make it behave differently, and so forth. And all this important information allows us to then respond and saying, is this a virus that has a variant with a concerning mutation that might affect our diagnostic tests, might fail therapeutic interventions, maybe we need to update our vaccines, and so forth. A lot of information can come out from this sequencing. Next slide, please. So taking a little look into our lab here at Biodesign Institute, where we are sequencing thousands of genomes of SARS-CoV-2 every week. We use high throughput sequencing. All these are robotics technology that allows us to process thousands of samples. This gives us very good coverage of what kind of cases are, what kind of variants are being circulating in Arizona state. We're not doing just small amounts of sequencing, we're really doing a large amount of sequencing here. Next slide, please. And the cases that we're sequencing of COVID primarily come from two areas. One, public testing, which is here in ASU, as Steve has mentioned, we have a clinical testing lab here that's responsible for a large part of the testing in Arizona. Every positive case from that lab comes up to us to be sequenced. And it's not, and the beauty of this is that we are right on site here at the Biodesign Institute. We're right in the next bench over from the clinical testing lab. And so that allows us to sequence these cases very, very quickly. Besides all the public testing, we also get samples from cases from hospitals around Arizona. And this gives us also a good sense of what are the kind of variants we're seeing that are putting people into the ICUs, severe disease, that sort of thing. From our sequencing, we then share this data back to the CDC, the Arizona Department of Health Services, and by tying it in so closely with public health, this allows us to respond to this, the different viruses as it evolves, update our policies and make changes, contact tracing, that sort of thing. So this is not just purely a research kind of curiosity. This is tied in closely with public health response. Next slide, please. I'm really proud of this here of our group at ASU, and that we are able to contribute to a large amount of sequencing in the state of Arizona. And secondly, we are at ASU essentially the leading edge of understanding how the virus is evolving in our state. The graph on the left shows you the amount of sequencing being done in our state. There are several labs that are doing that, such as TGen. CDC is also doing some sequencing for us. And the color in red, that's us at ASU. And we're doing a lot of sequencing here in Arizona. That is, we in Arizona here are sequencing our own Arizona cases. Secondly, you'll notice that the red bar is always about 2 to 3 weeks ahead of the other colors. And that is the value of being on the leading edge. We are able to give real-time information about what's circulating immediately, whereas many other states are not even sequencing or several weeks to months behind. And that information is not very useful. Real-time sequencing is. Next slide, please. And so here's a picture of the kind of variants that we can get this information from sequencing. It's a very colorful chart. I'll just walk you through that. Each bar shows you the variants of SARS-CoV-2 week to week from early of 2021 until towards the end of most currently in February 2022 to the right. The size of the bar color represents different proportions of the variant. And you can see that, for example, the blues are alpha variant, you might have heard of that. Greens are your delta variant, and then now red is the Omicron variant that we're dealing with right now. A couple of things that you can see from this is one, the colors change over time. That is because the virus is evolving, new mutations and new variants take over another. That's the reason why we need to continue to be vigilant in sequencing these variants, because the virus will evolve and we need to keep up with that and stay one step ahead of that. Secondly, notice how fast these colors can take over. If you look on the left side of the graph, the green delta took a couple of months to establish from nothing until becoming the dominant strain in the summer. Now contrast that to the right, the Omicron variant. You can see that it took over much faster than the green Delta. In a couple of weeks, from zero, it became almost dominant. That's how fast the virus can evolve. And that's why we have to be ahead of this virus and being vigilant in sequencing it all the time. And so if the third thing I'll end here as well is to look at the dates here. This is continuously, every week we've been sequencing that. And I've had the fortune to be working and leading a very talented group of researchers in my lab here, sequencing all throughout the year, even through the holidays. If you notice when the red Omicron first came up, that is right around Christmas and the New Year. So if you ask my lab, how was Christmas and how was the New Year? That was a time where they're putting in all the time to make sure that we had a good catch on how the virus was evolving. That was a critical time for the Omicron surge, essentially. I can even remember my Thanksgiving. That was the day the WHO released a notice about the new variant Omicron. So that was how I spent my Thanksgiving, planning for the next variant. So with that, I guess I'll open up to the group for questions, and thank you all.
Thank you, Dr. Lim. I think we all
Thanks, Efrem.
appreciate
That's great.
that. I do have one question. Are future variants expected to mutate and take over faster than Delta to Omicron?
That's a good question. It's hard to predict what the next variant will be and what properties it will hold. I think that if I was able to predict things like that, I would probably be in a different career, probably in Wall Street. Now, based on Delta, it told us that it can, between Delta and Omicron, the fact that Omicron could take over so quickly, so rapidly in a couple of weeks. Before that, I would say, there's no way it would take over that quickly. But our experience now with Omicron makes me pause and think perhaps we shouldn't be so quick to assume that. But regardless of what kind of variant is going to come up next, we'll be ready to catch that here because we're able to sequence it in real time.
Okay, thank you. So we have a couple of fundamental questions too, kind of going back to more basics, but how's SARS-CoV-2 kind of different from others, including like viruses that cause the common cold? So what are like the unique structural features that sequence confers causing disease?
Yeah, that's a good question. So SARS-CoV-2 is, in terms of respiratory disease, is on the upper end of being more pathogenic. Viruses like adenovirus, there are the coronaviruses that cause the common cold, and rhinovirus as well as common cold viruses. So some of these are completely different viruses, different families. So the minute we see those picking up those kind of virus, we can kind of guess that that person is likely has a more milder symptom. So the fact that they are very different in terms of the genome sequences, we can tell very quickly, is this a virus that's more usually seen in a severe case versus a less milder case?
Thank you. Melissa, I see you have a question.
Yeah, it's kind of a bit of a two-part question. One, can you comment on the stuff that's been coming out about the Omicron BA.2 variant? And tangential question to that, is there a reason why BA.2 hasn't been given its own Greek alphabet name and is just continuing to be referred to as Omicron Part 2, basically?
Right. Yeah, that is a great question. So what Melissa's asking about is that in the Omicron variant, we just call it Omicron, but actually there is a sublineage of Omicron called the BA.2, and that's its lineage name. That first emerged in about December or so, about January of 2022. That first kind of picked up in the US, and here in Arizona, we have been seeing cases of that as well. So far, they're holding about 3 or 4% of the cases that we've been sequencing. So it's fairly low so far still. That compares to other parts of the world where some countries are seeing a higher percentage of this BA.2 Omicron. Now, your question about why are we not seeing this kind of taking over that? Is there something different about this variant? Some studies are now emerging to show that although this BA2 variant has enough mutations that separate it from the original Omicron, it seems that people who have been infected with the original Omicron BA1, the immune responses from that seems to cross-protect you against the BA2 variant in that case. So in that case, perhaps in Arizona, the fact that the original BA1 Omicron had a huge surge, meaning most of the population probably had mounted an immune response against the original Omicron, would now be cross-protected against this BA2. And that might explain why so far BA2 has been holding pretty low in our communities.
Thanks.
Perfect. Thanks for that question, Melissa. I have a question from Josh. Why do viruses create new strains and how does it happen?
That's a great question, Josh. Why do viruses create new strains? That's what they do. Viruses don't have some ulterior motive of like trying a sinister plan of what kind of variants it wants to become. Every time the virus replicates, every new infection, as the virus replicates, creates an opportunity for mistakes to occur in its genome. These mistakes are therefore these mutations that we see. When enough mistakes are made, we then end up getting a new variant. An important part of that is this is an intrinsic property of the virus. Every time you get an infection, you're going to have chances of mutations. Meaning, the more cases we have, the more transmission of SARS-CoV-2 is out there, the more chances for the virus to make these new mutations and a new variant to come up. And so that's a natural property of the virus. I hope that answers your question, Josh. That's a great question.
Thank you so much.
Let me just quickly follow up. Do all the new variants become dominant if you get new variants?
Not necessarily so. We've had a couple of false alarms so far when people around the world are saying, hey, there's a new variant. Actually, Melissa was asking, why aren't we calling, a new Greek alphabet and so forth? And many of these things have come up. And not just a new variant, a new mutation does not mean alarm bells and it's going to take over. And we've seen that so many times. And that's perhaps one of the reasons why also BA2 has been held back from giving a new designation. We really want to reserve this new Greek alphabet as at one level, a global warning, call to arms to say, hey, there's something new that we should be worried about, we should be on alert for. And if, for example, this new variant that comes up doesn't really end up taking over or being a problem in the communities, then we don't need to be alarming the public about that. And so the answer is no. Not every new variant is a bad thing.
Josh, anything else?
No, go ahead. Sorry.
Okay, perfect. No, you're fine. I have a question from Mark Pearson. Are white-tailed deer a natural reservoir for SARS-CoV-2 in Arizona? And what about other wildlife reservoirs?
Mark, that's a great question. So that comes from a very recent study that's been released showing that in some parts of the US, they have found white-tailed deer harboring SARS-CoV-2 virus. We don't know how widespread that is so far, and it's not that all white-tailed deer are currently infected with that. To my knowledge, that study has not been done yet locally in Arizona. So the answer to that is probably we don't know that. However, given what we're seeing so far in that data, the fact that these are still one-off cases here and there, I don't expect this to be a major problem unless it fully passes through all white deer start harboring this virus and getting infected. When more of those cases, then that perhaps becomes a reason for concern.
Thank you so much. Is it true that viruses evolve to becoming more contagious and less lethal, or are there exceptions to this Darwinian rule?
Do they always have to be evolving to be more pathogenic or less pathogenic on the other end of that spectrum? Let me go back to again saying to the fundamental property of the evolution of the virus. it's just gonna, it's as it infects more people, it's gonna make mistakes. Does that mean it's always going to be more disease or less disease? That doesn't necessarily have to happen. For example, one thing that people have noticed is Omicron seems to be less deadly. So people come to this conclusion that therefore the virus is always going to be milder. That's very complicated and maybe a simplistic summary of that. But the reason it looks like it's milder is you comparing it to the virus before, where no one had any immunity. So we got the full brunt of the disease. Compared to 2 years later, everyone has some level of immunity, pre-existing immunity, vaccination, and so forth. So everything will always look better than the original 2020. Secondly, is it going to be always beneficial in that case? The trick again with Omicron is overall it was less deadly, but it infected way more people that a small percentage ended up causing a large number of cases. So then you've got now weigh in the balance of, this, how do we measure the burden of disease? Is it truly less deadly when in overall it caused a bigger burden on our public health system? Does that answer your question?
If I can add just one other comment on that.
Yeah, the way I ask the
Go ahead.
question, doctor, is that thesis from virologists is that in order to survive, the virus gets less lethal in order to be, that's kind of the theory I'd heard. Is that, and that's why I was asking whether there was a general rule or not.
I think you got cutting out a bit, so let me try to repeat your question if I got it correctly. I think you're asking, is it true that the viruses always try to be less pathogenic in order to be able to continue
survive,
transmitting and survive out there?
To survive, obviously, if it kills people, it can't replicate as much. Yeah.
Right. That is a possible outcome. On the other hand, I would like to point back to my experience of SARS in 2003. It was very deadly, and we eradicated that. So there is still a world in which you can still eradicate these viruses. Over these cases like that, it's hard to predict, and saying that therefore this is a long-term end game of the virus always trying to be endemic is I think where you're going to be mild, and no one really cares about that. Mm-hmm. But that doesn't have to be the case.
Yeah,
thank you.
Just to add to that, JP, I think you hit the nail on the head. The goal of the virus is always to replicate, right? The goal of the virus is to infect as many people as possible because it wants to reproduce itself. It doesn't really care whether it makes people sick or not, but obviously, if it makes less people sick, then there are more people to infect. So you would think that that would be true. It is important to remember that there are plenty of viruses that over time have not seemed to get much less pathogenic. And so, HBV and HPV and, any number of viruses over time still are pretty bad for us. So it doesn't always happen that they get better.
Thank you. Thank you, Josh. Thank you, both of you.
I have a question, Efrem. So what will you do with all the sequencing capabilities after COVID? What are your plans for the future?
Yeah, that's a good question. Obviously, we can sequence SARS-CoV-2 really well, but that's not where we're going to stop. In fact, what I think we've demonstrated here is the value of being able to keep track of all these infectious diseases using this technology. And so we are preparing to expanding this towards other infectious disease pathogens, surveillance in our communities. One thing we're really excited about is in wastewater sequencing, because wastewater essentially can sort of give you the health status of a community level. Even if those individuals don't get tested, by surveillance through these wastewater community systems, we can figure out what kind of infectious diseases are circulating in communities and respond accordingly. Where I could see this working really well, for example, are in nursing homes or in campuses. And all we have to do is to maintain this surveillance, sequencing all these viruses. And when something unusual comes up, we can quickly step in and start responding to these threats quickly, even before we wait for people to go get tested themselves. So many things we can do with sequencing, even past COVID.
Thank
Dr. Lim, I still have some more questions.
Sure.
When will an Omicron variant booster become available, or the next booster, perhaps?
I don't have any information on timeline, but I know for a fact that many of these other companies are developing that Omicron booster and other combinations. I suspect that's going to be needed because the vaccines that we have right now were based on the strain 2 years ago that we don't see currently circulating and haven't seen for a long, long time. So it will come. I don't know when.
What kind of information do they use to make, like sequencing wise, to use their justifications in making their next booster? Is there an insight that you have on that?
So what we obviously want to do is be able to predict what the next variant is, and that's difficult. And if not, once we detect a new variant that is going to be a concern, quickly get that into the queue in the pipeline to update our vaccine. So in terms of the second strategy, I think that's the better route, meaning that we have done that, for example, for flu. Now the time scale is a little longer. Every year we have an updated seasonal flu vaccine. It's based on information of what's circulating around. We have way more resources now in for COVID-19 that we could do that same thing. Look at what's circulating, quickly fast track through that into our vaccine productions and update that quickly. So I feel that that's a possible route forward with enough resources.
Great, thank you. I have some more questions. Good thing we have some time. Let's see here, sorry about that. As respiratory infection season ends and COVID-19 rates continue to hopefully decrease, but in theory, do you expect a new variant surge will occur before fall?
Whew. Again, asking me to predict the future, yeah. Let me first start saying that, okay, I'll start with where I think, and then I'll end with a positive note, because I think, there's me, the scientist, about what I think based on the data is going to happen, but I also don't want to leave you there hanging and say this is all doom and gloom, okay? So I'll first tell you what we know. What we know is that our immune responses against coronaviruses, including SARS-CoV-2, decreases over time. And that window of time takes a couple of months, 4 to 5 months or so, meaning that we have just gone through this large Omicron surge. A lot of people have now immune responses against that. But given that timeline, and if immune responses, this happens naturally to everyone, immune responses just drops over time from coronavirus infections, that puts us into somewhere in summer. So the key is going to be what happens in summer. If in summer, around the world, the global incidence of coronavirus is low, will be unlikely that we'll see this problem of a new Omicron, a new variant surging and so forth. Why? It's because the same thing, the fundamental thing that Josh was asking about, how do viruses mutate? How do we get new variants? The more cases there are, the more chances for a variant to come up. If we bring this all down to a low, manageable level, chances are we will not give the virus the chance to mutate a new, escape variant or so forth. So that's based on the data that we have so far. I'm on the team of being optimistic, meaning that the glass is more than half full. We have been through this so many times. We have better vaccines, we have better antiviral drugs. We know what we're doing right now, and we don't have to live through another surge. And that's what I'm banking on.
Thank you for that. Another question from Mark. Any chance of, oh, sorry about that. Looking for Mel. How widespread is the sequencing technology around the world?
That's a good question. So the sequencing technology that we use here, it is quite sophisticated. And And as you can see, there's a lot of benefits to it right now. The good thing right now is that a lot of countries around the world have stepped up the infrastructure of sequencing in order to support that. So much more sequencing is occurring now than at the start of the pandemic. However, there are still many parts of the world where we still have overall low levels of sequencing. And so, yes, we're doing better, but we're probably not on a global scale, not at the level that we need to be. And there are many people and many organizations trying to beef this up, to beef up the infrastructure and all that. And that's more to do with the inherent disparity and low resources of some of these countries. And so that's one area I could see us doing better as a global community.
Perfect. And then kind of to jump off from that, I know we have a lot of capabilities, but could the COVID experience, at least here in the US, also improve other vaccines, making the flu vaccine more accurate?
Exactly. That's the whole idea here. We don't
Okay.
want us to just be a blinders on just SARS-CoV-2 and COVID and all that. This is not a new problem that just came on our plate. We here at Biodesign have been thinking about these infectious diseases for many, many years now. And what we should do is to leverage on all this experience, this huge wealth of experience here and expertise here, and broaden that to other infectious diseases.
Thank you so much. I want to open up to the floor. If anybody wants to unmute themselves and ask an additional question, I'd love. These questions are great. Thank you so much. Let's see, anybody? No?
Do you want to, I know you pointed out that the immunity to infection goes down pretty quickly. What about the immunity to severe disease? I don't know if you want to comment on the differences between kind of B cell and T cell and that sort of thing.
Right. So Josh is making a good point about the immune responses. In general, there are two arms of the adaptive immune response. One is called the T cell immunity, one's the B cell immunity. So most of the things we're talking about in terms of antibodies, that's the B cell immunity. Whereas what we have not talked a lot about is T cell immunity. They work kind of hand in hand, where essentially the B cell immunity is kind of like your first line defense. These are antibodies that are going to block the infection. The T cell immunity is your sort of like your second line of defense that would clear out infections, essentially. A lot of the variants that we're seeing here seem to be evading the first line of defense, the antibody responses. That means that people are getting infected. However, our T cell immunity is holding strong so far. It does not seem to be failing us too much. What that means is that it protects you from severe disease progression. And that's the reason why people should still get vaccinated, to get your T cell immunity up there. And so, for example, everyone's complaining, oh, I got vaccinated, but I still got infected with Omicron. Yes, because Omicron evaded your first line B cell immunity. But notice, you didn't end up in ICU. You didn't get severe disease. That's because your vaccine provided your second line T cell immunity. And that's really important to remember that, so it's not just infection is the only thing we care about here. The fact that you didn't get severe disease, that's a win. That's why we should all get vaccinated still.
May I ask a question about long COVID and related to a last answer here, the role of the immune system, or whether it is more a question of respiratory system going amok.
JP, we're having a hard time hearing you. You're coming and going. You might want to type your answer.
Oh, I'm sorry. Yeah, so very simple. Do we have an understanding of the mechanism of action that leads to long COVID, whether it's related to an immune response gone amok or some other reason?
Yeah, so I think JP's question is asking about the long COVID part of SARS-CoV-2 infection and what's the role of the immune response in terms of that. Let me first start off saying that we have an abysmal knowledge and understanding of long COVID. we've been in this for 2 years now, and we barely know much about long COVID. This is a huge problem that's been kind of overlooked and needs to be studied taken very seriously given the amount of infections going on. So the problem is we don't have too much knowledge on what's going on there. It's multi-tropic, meaning that there's multiple symptoms associated with long COVID. It's not clear to us, to be honest, about what's actually causing that. Why are some people still having these symptoms and disease many months after the initial viral infection? Maybe Josh might be a better person to chime in on that, Josh.
You know, it is complicated. I think Efrem got it exactly right. First of all, there are different versions of it. So some people have long-term respiratory symptoms. Some people have CNS, so brain symptoms, something called brain fog and memory issues. Other people have GI symptoms. In many cases, people who get this have had not necessarily severe disease. Many of them have never been in the hospital for COVID, but months later develop these symptoms and they persist a long time. And JP, you'll of course appreciate that some of these things are not dissimilar to what's been seen in other infectious diseases where there have been long-term effects. One observation that people have made recently is the presence of autoantibodies. So this gets at your question about the immune system. People seem to be making antibodies against self-proteins, and that may play a role. We don't really know yet. But Efrem's exactly right. You know, there's really not very much studying going on. In fact, several of us are trying to get samples, and it's very hard to get samples from people who have long COVID in order to study them and kind of see what's different, because we'd like to look at the immune system. There may be secondary infections, there may be persistent infections. We just don't know.
Yep. And
yeah, just a quick one on this. One thesis, and it's only very, very early, is the role of underlying infections, pre-existing infections like EBV and others. Yep. That may help create that longer, but that's only an assumption. So I don't know what you guys think.
I don't think we know. I think we don't know yet.
Right, and there's a lot of precedents for that, meaning a pre-existing infection and then a second hit coming in, kind of recurring, re-triggering it. That model has been known before. And besides the samples, we've been trying really hard to try to get the funding to do this. Where our lab is looking at is the GI aspect, gastrointestinal aspect of long COVID. And for whatever reason, people kind of only focus on the acute respiratory disease. And it's so hard to get people to let us study the long COVID. And as more and more people get infected with COVID, this long COVID is going to be a bigger problem in the long run.
Right.
Thank you.
Dr. Lim,
Susan's asking about EBV. So that Epstein-Barr virus, that's a herpes virus infection that can be a prolonged infection, sometimes lifelong reactivation and so forth.
It's the virus that causes mononucleosis, for those people who are familiar with that. But it has also been documented to cause a couple of different cancers and other things. The vast majority of adults in the US have been infected with EBV. In our studies, probably more than 90%.
Dr. Lim, I had another question about jumping species. So if this virus has non-human reservoirs and can jump between species, is there a greater chance of a more pathogenic mutation than a virus that tends to just remain species specific?
So what happens when SARS-CoV-2 goes into a different animal reservoir, and then should we be worried about it coming back into humans? That has happened before. So the minks, for example, at one point of time, the virus jumped into the mink population. You know what we did when we found out it was in the mink population? We culled it. Do we see mink viruses coming back in humans? Nope. But meaning that there's a solution to that. On the other hand, it is not always when it goes into one host, it can then freely jump back. In fact, in terms of virology, there are many instances in which once it gets to a new host, it kind of adapts to that new other non-human host so much that it doesn't do so well when it comes back to the humans. So it does not always have to be that case where it just kind of flip-flops between humans and some other animal reservoir like that. That's a really good thing for us.
Good news. Any other questions? I did my thesis, Melissa. I did my
Yeah,
thesis.
just
Go ahead.
comment in the chat. You guys mentioned that you are having a hard time getting samples from people with long COVID, and I was just kind of curious what the bottleneck there is, because some of the people that I work with on my long COVID and policy thesis are very enthusiastic to participate in research, and I'd be happy to put you in contact with them if you're looking for people to participate.
That's helpful. You may hear from us.
Yeah. Also, Michelle Villegas Gold. She's, if you know her, she's a good contact person.
Yeah, no, we're working with her already.
Cool.
Yeah.
All right, we have time for one more question, and then we're going to wrap it up. If there is a last question anyone wants to ask.
Where'd it come from?
We don't know, and I don't think we may ever be able to find out. But it is for sure not some lab invention that some nefarious person released it out. No.
Yeah, our colleagues at the University of Arizona have a very nice paper recently, really showing that this outbreak really came from a market in Wuhan, where they sold animals. Yeah.
All right. Well, thank you very much for an excellent talk, Efrem. And with that, I'd like to turn it over for a couple of minutes to our colleague from the foundation, Elizabeth Munkel.
Thanks, everyone.